the mothers who are here are welcome to write their questions in the chat box and we will take them one by one. So feel free to ask all your questions and doubts. And until you're typing, I shall start with the questions we have received on our support groups. Welcome, Dr. Abhijit. I will start with the question answers that we've received on our support group. ReeLabs has a support group by the name of Janani, where we invite pregnant mothers to be a part on the WhatsApp group and we help them through the journey of their pregnancy. And some of the questions that we regularly get on stem cells, I shall be asking you today, Doctor. First and foremost, Doctor, is in case a pregnant mother misses out on the opportunity to bank during the delivery of a baby, what are the options that are available later, and are they as good as what cord blood stem cells are?
Yes, I got the question. Again, apologies for the technical glitch. The question is, in case any mother has missed out on banking these precious cells, what are the options available? In fact, all of us have missed out. I mean, I'm talking about my generation, I'm talking about the next generation. So at the same time, this is a very evolving field, a lot of action happening, a lot of work happening, a lot of therapies using stem cells coming up. So it's obvious that people who missed the bus should not be left out. Thankfully, there are many adult sources, which means you can bank stem cells from your own body from various sources. And the ones that are commonly used in practice these days is the bone marrow, the dental pulp. That's the pulp, especially in the permanent teeth. Also, hair follicle. We're looking at adipose tissue or fat. A lot of people undergo plastic surgery or liposuction. So rather than wasting or rather than throwing away that fat, it's a good idea to preserve stem cells, which are a very rich source of stem cells. Also, menstrual blood in women. These are all very good sources of stem cells, and they all should be looked at in case you're looking to bank it.
Thank you, doctor. And could you help us understand why is it that cord blood stem cells from the placenta, how are they, why is it that, the technical people who are respectable, notable people from the field of stem cells, why is it that they say that placental stem cells and cord blood stem cells are superior? Why are they called superior? Can you help us understand? Because these terms are thrown at you when you're pregnant, but we need to understand that why are they called superior stem cells?
Basically, I just mentioned there are plenty of sources of stem cells. And to make it simple, there are two kinds of sources. One is the products of conception, and the other source are adult stem cells. I've just given options of adult stem cells in the previous question. With respect to the sources of stem cells in the primitive age group, we're looking at maybe cord blood, cord tissue, placenta, amnion, amniotic sac. These are very rich sources of stem cells. I've been asked and I've been put in a spot plenty of times to pinpoint exactly which is the best source out of these five, which you can see on the left of your screen right now. Strictly going by research, strictly going by the quality and quantity of stem cells, the placenta is definitely one source that stands heads over heels with respect to any other source of stem cells. The reason being, firstly, placenta is a very bulky organ. It's almost a 1.5 to 2 kg organ. Secondly, it's very, very rich in stem cells. It's got a huge quantity of stem cells, obviously because it's a bulky organ. And more importantly, even in terms of quality, there are some kinds, each stem cells, they possess something called the biological marker, which determines how the stem cell behaves. So placental stem cells have got basically the markers of primitive, embryonic, and adult stem cells, which obviously means that maximum number of therapies, if attempted or instituted using placental stem cells, will yield the best possible results. Again, this is as per research. One glaring example I can right now speak about is the fact that we used placental stem cells in India's first and only stem cell based clinical trial against COVID-19. We did this using bank placental stem cells. It's an FDA and a DCGI approved clinical trial, and we did it in Sion Hospital in Mumbai. We treated moderate to severe patients of COVID, not patients on ventilator. And as we all know, patients in COVID, we've all lost somebody. They've all, generally the death or mortality in COVID happens because of cytokine storm, which means your own immune system goes haywire, it becomes hyperactive. Stem cells from placenta are immunomodulatory. They regulate your immune system so that the growth factors and the cytokines come back to the baseline levels. So they did that with the patients of COVID-19. That is how all the growth factors came back to normal, and instead of any mortality, the patients were benefited. They were completely cured and discharged. So that is why, giving an example of COVID, which is a very recent addition to the clinical disorders in or the clinical spectrum, placenta basically has a role to play in essentially almost all disorders across the board. And in case there's an option of banking placental stem cells, it's a good idea to seize it. Do not let it go waste. Thank you.
Doctor. Doctor, another commonly asked question is that what is the process? Okay, the person has delivered her baby. When is this process done? Is it done? Where is it done? When is it done? How long does it take? Does the mother or the baby feel any effect of this collection of the sample?
Firstly, this is a very, very safe process. It's a very, very convenient process. And most importantly, there is no harm whatsoever to the mother or the baby. Let us first understand, I'm a doctor first. The most important thing during pregnancy and subsequent delivery is optimum health of the mother and the baby. That cannot be compromised. That should not be compromised. And it will not be compromised. The fact remains that with respect to cord blood collection or collection of placenta, it happens only after the complete safety and well-being of the baby and the mother is established. To give an example, after the baby is delivered, we all know that the cord tissue or the umbilical cord is something that connects the mother and the baby. What needs to be done is, I think in any routine delivery, the cord is clamped. And after it's clamped, it is then the baby is separated and given to the nurse for cleanup. Only after that is done, that is when the cord blood is collected. Okay, so basically the baby is already separated. It's out of reckoning. So there's no problem with respect to needle pricking the baby or the mother. The baby is already separated, and the process of collecting the blood only happens after that's happened. The needle is inserted in the umbilical vein, which is a part of the umbilical cord. Again, it's absolutely painless. The mother will also not feel when that happens. Once the cord blood is collected, we then wait for the second delivery to happen. That is the delivery of the placenta. That also happens organically. So we just wait for the placenta to be delivered. Once the placenta is delivered, it is then collected and stored. So it is a very safe, to repeat, very safe, convenient and effective process with zero harm whatsoever to the mother or the baby. And the potential benefits of banking stem cells from the placenta, cord blood cannot be underestimated. Thank you.
After the collection, doctor, in how much time, suppose somebody is banking from our lab is based in Bombay, some labs may be based in other cities. How much time does a person have after the sample is collected for it to be processed and stored in optimum conditions? What is the window that we have?
Yes, that's a very good question. It's basically this entire process is dependent on banking a biological sample. That is, whether it's cord blood, cord tissue, or placenta, or amnion or amniotic fluid or amniotic sac. These are all biological materials. And biological materials will definitely have a shelf life, whether we like it or not. With respect to cord blood banking, it's a licensed activity. There's an FDA license that is given to cord blood banks, and they are continuously regulated. Surveillance happens, and everything with respect to the SOPs, processes, the the lab personnel, etc., checked by the regulators from uh, 72 hours on the higher side to begin processing. However, there might be occasions when it can be collected even beyond 72 hours, which is not something I recommend wholeheartedly. Simply because just consider something like a vegetable. Supposing you just take buy an apple from a store, from your grocery store, or uh, from your fruit vendor or vegetable vendor. Just leave it as it is. The first day it'll still be okay. Supposing it's a ripe apple, over a period of time, it is bound to deteriorate and turn rancid, and then eventually we'll have to discard it. Biological material also works on the same principle. The maximum freshness, or the maximum, and if I can be scientific, the maximum quality and quantity of stem cells are there when the process can be started as soon as possible. So though the FDA has given a window of 72 hours, it's a very good idea to always bank with your local bank, and as soon as possible, the processing has to begin. For the processing to begin quickly, the sample has to reach the lab as soon as possible. I recommend anything between 6 to 10 hours or maybe 12 hours for the sample to reach the lab and the processing to begin, to uh, ensure optimum quality of cells, both in terms of viability, potency, and count. So if that can be done, we can be rest assured that the maximum goodness from your biological sample has been preserved. So the watchword should be earlier the better. Though there is a 72-hour window that the FDA has given, I would like to keep that on the side. Fine, that's something that the regulators have said. But like any biological material, it is imperative that the cord blood bank or the stem cell bank that takes your sample begins processing as soon as possible. Anything between 6 to 12 hours is good enough. Thank you.
Thank you. What are the options available to pregnant parents with respect to the number of years they can freeze the sample?
I think the question was, how long can the stem cell sample be banked? Is that correct?
Yeah, what are the options that are available? As in, what is recommended? What are the options available? And what is actually possible? What are the possibilities in the future with respect to storage?
See, I think most cord blood banks have various packages with respect to time. The packages can be anything between 20 years to 75 years, if I'm not mistaken. 75 years because it's considered a lifespan of a person. Does not mean that after 75 years the cells will wither away or die. The cornerstone out here to be recognized is, once the processing is done and stem cells are preserved, they're preserved in something called liquid nitrogen. Liquid nitrogen and the gas that is emitted by this liquid nitrogen, which is gaseous nitrogen, has this power of putting cells to sleep. And when these put the cells to sleep, it also means that the cell which is put to sleep can be put to sleep for n number of years. There might be packages for 15 years or 30 years, 40 years, 75 years, whatever period. But the cells, once put to sleep, can be kept in a sleeping condition for n number of years. So even beyond 75 years, if they are kept in the sleeping condition, that is in this case a frozen condition, the cells will retain their viability, potency, and count till they are thawed. Now, the question that was asked was, how long should one bank? Again, I'll go back to the fact that this is a very nascent field. However, a lot of work is happening as we speak. It's possibly in the biotech field or the biomedical field, the fastest growing activity, that is a stem cell activity. And considering that the first cord blood collection happened in 1988, okay, we are just a 35, 40-year-old industry. And right now we are talking about the first line of treatment for so many disorders in the bone marrow, right from leukemia, thalassemia, multiple myeloma, lots of cancers, a lot of research happening in cerebral palsy, in Alzheimer's, in Parkinson's, in autism. I mean, there's possibly no disorder or no system which will be left untouched by stem cells. And the option of using these cells is only there once you have those cells ready or once you have those cells handy. So it's a good idea to bank for as long as possible, simply because you never know when the stem cells that you've preserved will suddenly become the first choice of treatment or the first line of treatment for a disorder that had no treatment. So in case you don't have it, there's no way you can use it. So it's a good idea to bank it for as long as possible, especially considering the rapid strides in research that stem cell industry is making. Thank you.
Thank you, doctor. In case any of the parents who are pregnant families that are attending today have any questions, you can either unmute and ask or leave your questions in the chat box. Welcome your questions, your doubts, your queries. Dr. Abhijit is the best person to be able to reply to them. While we are waiting, I have a few more questions that have come to me through the community, through our support community. Somebody wants to know that there are some stem cell banks which are offering community banking. What are your thoughts on that? And could you help us understand what the concept of community banking is?
Community banking essentially means that the sample is kept or preserved for the community. So the term community banking has got two words, community and banking. So if both these words are sincerely looked at, then this entire exercise is extremely noteworthy and extremely meaningful. So what are these two words? One is community, one is banking. So with in the stem cell field, a community is considered millions of samples. So in case you have millions of samples in the community and all of them have signed up for community banking, then it's a good idea. Secondly, assuming you have millions of samples in the community, now I'll give an example of NetCod. NetCod has got, which is like the International Stem Cell Registry, it's got close to, I think, what, 20 million, if I'm not mistaken, that was the last count of stem cell samples. And what they've done is they've diligently done the HLA or the genetic status of all the samples that they've preserved. So just having a community is not good enough. You also need to get an HLA done. So when any stem cell bank advocates community banking, the first question you need to ask is what is the size of your community? Few thousands, few lakhs will not make any difference, I promise you. Secondly, assuming that you're ready to bypass that first hurdle and look at community banking, even with a few thousands of samples in the community, the next question that you need to ask is, has the HLA been done for all these samples? Because unless the HLA is done, there's no way you can take anybody else's sample. Because HLA is something that determines your immune system. And depending on the HLA match, it determines whether somebody else's sample can be used for your disorder and vice versa. Now, I'm also a pathologist. An MD pathologist also have multiple laboratories, blood banks. I know the costings of HLA. It costs a few thousands of rupees as cost price. So even with all the subsidies, it's still a few thousands of rupees that will be have to be shelled out by the cord blood bank or the stem cell enterprise that is advocating community banking. So in case that has been done and everybody's HLA has been made and made public, so that you as a customer can have access and you can see it. This entire exercise is nothing but a big disaster, a big sham. So unless you see that yes, it's a big community. Secondly, even if it's a small community, everybody's HLA has been done and has been displayed for everyone. There's no way this concept can work. I'll just give you basic mathematics, a few thousands into a few thousands. I mean, assuming a community of say 50,000 samples into even 5,000. the amount is huge. Any cord blood bank that actually does this will no longer be a cord blood bank. It would have possibly gone bankrupt. So that's not possible commercially unless it's a public bank. So private banks cannot do community banking, and it is not advocated because it's not a service. So again, there are two things in the community banking. Number one is the community needs to be large. If it's small, avoid. Secondly, even if it's small and if the HLA is not done of each and every sample in the community, big avoid. And thirdly, most importantly, once you pledge your sample to the community, it means that you have no access to it. Let me give you an example that when we bank any cord blood sample, that sample belongs to you. It's like a bank. We are only custodians. Once you bank your sample with us, we are supposed to preserve it for you. Tomorrow, you want to go and register yourself in Netcord. You want to auction your cord blood to anybody, maybe in maybe Russia or US or wherever, for millions of dollars, which actually happens. Again, I don't want to get into the commercials, but cord blood samples are auctioned. That is entirely your prejudice. That's entirely your prerogative. Nobody, and I mean nobody, should have the right to try and jeopardize it. So by pledging your sample to the community, you are depriving yourself of an option, which could be an income option, may or may not be something I subscribe to, but that's besides the point. But there is an income option in case you want to give it out to somebody who needs it. So that too has been sabotaged. That too has been tossed out. Hence, because of all these reasons, I do not advocate community banking, and I only advocate community banking if it's done the right way, which in this day and age is almost impossible to implement. Thank you.
Thank you, Doctor. Doctor, could you help educate the pregnant parents on the group regarding delayed cord clamping and how stem cell cord blood banking is complementary to the process of delayed cord blood?
A delayed cord clamping is basically a process by which maximum cord blood is sent back to the baby. I'll go back to what I mentioned in the very first question, that the health and uh, well-being of both the mother and the baby is most important and cannot be compromised, should not be compromised. So, uh, retrospectively, I also mean that anything that could be done at the time of delivery to ensure maximum benefit to the mother or the baby is something that should be given a shot to. In this case, there have been plenty of activities and plenty of research that supports delayed cord clamping simply because they want maximum cord blood to be given back to the baby. And there is research, I mean, which is a little uncertain, that says that this cord blood, once given back to the baby at the time of delivery, may benefit the baby with respect to, his health, well-being, immunity, etc., etc. Again, anything that can even remotely benefit the baby should always be something that we should look at. Hence, if there's any meat or any metal in this narrative, it's a good idea to do delayed cord clamping. Now, do I support it? Answer is a big yes. I do support delayed cord clamping simply because anything that can benefit the baby is something I'll support. However, unlike what people believe or unlike what the general notion is, in case you believe in delayed cord clamping, you cannot collect cord blood. That is incorrect. Okay? Because whatever said and done, there will be some cord blood that is remaining, a residual cord blood that will be remaining in a placenta. So, if I give an illustration, when we talk about delayed cord clamping, we look at waiting for 15 to 30 seconds, maybe a minute, before you clamp the cord. But whether we like it or not, there will be a time at some stage during the delivery process that you need to clamp the cord. And assuming you've taken a minute, so that maximum cord blood could be given back to the baby, even after a minute, you will clamp the cord and then separate the baby. The process of cord blood collection comes in after the baby is separated. Again, I'll repeat with a capital AFTER. After the baby is separated, whatever is left in the placenta is good enough to bank simply because you will still get an adequate count. And in case the count is inadequate for whatever reason, there are options that we have worked on, which is called in vitro multiplication or in vitro amplification, where the count can be enhanced or amplified in the laboratories, so that we can get a count that is optimum for to facilitate a successful transplant. So I support and vociferously support delayed cord clamping. All I'm saying is, after the baby is separated, after doing delayed cord clamping, whatever blood is left, it's a good idea to preserve it. And in case the count is low, you can always, in fact, the cord blood bank, like me, I would, in our case, since we have the technology, we'll get in touch with the customer. We'll tell them that, listen, the count is for whatever reason low, whether it's because of delayed cord clamping or because the quantity that was collected was insufficient or XYZ reasons. Whatever the reason is, in case the cord blood sample is insufficient, there is a option, there is a plan B where we can expand the cells. And after we have brought it to a particular uh, standardized level, that is when we can preserve it for future use. Thank you.
Doctor, we'll take the question that has been put in the chat box by Supriya. Can you please share any example where stem cells were used to cure disease in child or parents?
Supriya, there are plenty of examples, and if we start talking about examples where this has been done, I think we'll require many more sessions. But I'll try and make this easier for you by just saying that right now, stem cell therapy is the only option for any bone marrow disorders. Okay, so that is the treatment of choice. So whether it is leukemia, whether it's thalassemia, whether it's sickle cell anemia, whether it's aplastic anemia, whether it's multiple bone marrow cancers, whether it's multiple myeloma, whether it's myelodysplastic syndrome, and these are all, I know they all sound Greek and Latin beyond a point, but these are all quite commonly seen bone marrow disorders. As per the recommendations of the regulator, this is the first line of treatment for these disorders. Secondly, so that's 100% cure. So to answer your question, straight away there's 100% cure. Secondly, the strides that stem cell industry is taking in various degenerative disorders is gigantic. There's a lot of work, and I just gave an example of something that we did. We did some work in COVID-19, which is a disorder that most people heard of for the first time 2 years back. And stem cells, being stem cells, had a role to play even in that disorder. Besides, we're also doing, and again, it's been the trial got completed, the patients benefited, they've all been discharged, and the results have been published in what we call the Big Daddy of Stem Cell Journal, that is American Journal of Stem Cells. Also, we are doing clinical trial on diabetes and diabetic foot, where right now the only option is amputation. So this is limb salvaging, can save somebody's limb. We are doing some work on pulmonary fibrosis. Again, all these, when I'm saying doing work, we are talking about DCGI, FDA approved clinical trials, because that is the only way that you can showcase the mettle of any therapy. The only way it's to be done is supposed to be done with a clinical trial, show the benefits, and then approach commercialization or market authorization. We're also doing some work on liver cirrhosis, especially alcoholic liver cirrhosis. I cannot overestimate the impact of alcohol in any country. In India has a dubious distinction of being a capital of alcoholic liver cirrhosis. So this is something that can reverse liver injury and can preserve life, save life. And people who are waiting for a liver transplant, will give them valuable time to in case there's no other option. So apart from that, there's work in Alzheimer's, there's work in Parkinson's. I know for a fact that stem cells have been used in autism and cerebral palsy in pediatric age group, where there were children who were going to special schools, and I have an example, that there's a publication on that I have personally given, where a child who was diagnosed with cerebral palsy, the parents decided to have a second child so that they could use the stem cells of the second child to treat the first child. And the first child who was going to special school is now completely cured and going to a regular school. So there have been all these, breakthrough news articles and activities that stem cells have been able to establish and showcase. So definitely stem cells is something that we should look at as a first line of treatment for many disorders. And I can tell you one thing for sure, that within 10 years, With all doctors, we will consider stem cells first as a line of treatment and then look at something else. So hope that answers your question, Supriya.
Mr. Ronak Ray wants to know, are the stem cell treatments safe or 100% proven to cure diseases?
I think I already answered most of it in the previous question. The reason being that I've given the fact when I mentioned that uh, stem cells for a multitude of disorders is the first line of treatment. When I say first line of treatment, it means that it has been regulated and uh, permissions have been given by the regulator for stem cells to be used for a bunch of disorders. There are almost 80-90 disorders which can be treated only with stem cells. And when I'm saying it can be treated, which means it can be, and the condition can be cured. Of course, if it can be cured, it's absolutely safe. And it is something, in fact, not, I'll go one step further, not only safe, but the only treatment modality that is effective in reversing the condition. Thank you.
Thank you, doctor. Another question that we've received is that if I've banked for my first baby, do I need to bank for the second one? And a similar question that you can take together is, in case of twin pregnancies, do you bank for both the babies?
We've got these cases. I have plenty of illustrations. Typically, in case there are two separate placentas, then they are not identical twins. Okay, and if they're not identical twins, it's a good idea to bank both the material, simply because you're firstly enhancing the option of using it for yourself and your family by 2 times straight away, because they will match each other. In case X needs a Y's cord blood or Y needs X, I mean, your X and Y are like siblings. In case both of them are available, there's never going to be an exhaustion of the material. There'll always be adequate material available. And most importantly, with the rapid strides that stem cell industry is taking, again, I'll go back to the fact that I don't have my own cord blood that is preserved, simply because there was no cord blood banking when I was born. And most of the adults here, I'm sure none of you had even an idea of what cord blood banking was till recently, and especially when you all got pregnant and were looking at options of cord blood collection. So it's always a good idea to have a very high number of cells available at your disposal. So in case it is required, you'll never fall short of it, because the rapid strides that this industry is taking, it's inevitable that it will have a role to play in many disorders. I mean, again, I'll give an example of diabetes. Something like diabetes, almost it's 1 in 3 or 1 in 2.4, if I'm not mistaken. So in case stem cells can be a treatment of choice for diabetes in say 3 years, just giving an example, maybe it could be 3, 5, 10, whatever years. Unless you have adequate cells, there's no way you can treat it. So it all starts with the fact that if you have an option of banking it, don't let it go. Because all of us, I mean, me included, we didn't have that option. So we had to look at other sources, which are not as good as stem cells from the pediatric source or the products of conception. We look at clutching to straws by banking our bone marrow, by banking our fat and all, which, and let me tell you, the quality and quantity of stem cells from any adult tissue is nowhere close to the kind of quality and quantity of stem cells you get from any source at the time of delivery. So in case that option is available, do not hesitate, just bank it. You never know when you require and you will still fall short. Thank you.
Thank you, doctor. Are there any other questions? Because I have my last question before we end the session, which is that some cord blood banks are offering dual storage. Can you help us understand what dual storage is? Is it really needed, or is it more of a publicity gimmick?
Dual storage essentially means by storing at two separate locations. And that is my understanding. Correct me if I'm wrong, because there might be other connoisseurs here. Uh, and they say that it's a good idea to store at two separate locations, because if there's an earthquake that hits one location, or there's some act of God that hits one location, at least uh, do you have some other location to fall back on. I really don't understand what the uh, rationale in this is. Because if there's an act of God in one particular location where maybe you stay, you don't stay, and it will cause devastation enough to possibly uh, a lot of lives, then I mean stem cells preserving it or not preserving it beyond a point becomes redundant. So I really don't see any major benefit because the chances of an act of God are almost negligible. Of course, there are there, but let me tell you, at the time of giving a license, FDA is very, very clear about a few conditions. For instance, I'll tell you what we have done. There is, if anybody goes through the FDA manual of what a cord blood bank needs to have and what it doesn't need to have. First of all, it needs to be in a green zone. It needs to be in a low earthquake gradient of sorts. There are some parameters that have been laid down. Why? Because they want to be 100% sure that even in case of an earthquake or a flood, the facility would be absolutely safe. So any cord blood bank that has a license, that is the first thing that they will look into, and only then they will give the license. So any cord blood bank which has a license at this point in time are basically safe to store with. Don't worry about a single or dual storage. But to extrapolate this question, I'll go into further details of bag versus vials, because that is something that will really mean much more than dual storage. Uh, in bags you can only collect in one or two bags, and basically if it's required, that bag is picked up, thawed, and given out. So it's over. It's your cells are over. Supposing you've stored say 100 million cells and the bag has 100 million cells, and for that disorder you only require 10 million cells. Since it's all stored together, once you thaw it, you'll use everything, and it's 90 million will just go waste. So I would suggest, if there's an option, bank in vials, simply because what happens in vials, there are multiple options. There are stem cells stored in multiple vials. So that 100 million of cells, I mean, again, 100 million is just an illustration, does not mean that it is exactly the count. Assuming the count is 100 million, it can be stored in 10 vials of 10 million. So in case a requirement comes up, only one vial can be picked up. The 9 other vials are still frozen and can be used subsequently, can be used for some other condition, or can be used for some other patient as and when required. Again, I go back to what I mentioned initially. Give yourself the best chance of usage. The whole premise of cord blood banking and stem cell preservation is, if required, you should have them handy. The best option of having them handy is in case you store in multiple vials, so that depending on whatever number of cells are required, only those many vials are picked up and used. The others are kept for future occasion. Hope this answers your question.
I'm sure it must have helped people understand what dual banking is, dual places, banking in dual.
And as we speak, before we wind up, I'm seeing two more questions.
Just
One
one
is,
second, doctor. There is
let's tell
somebody, there is Xiaomi 210911DI who is raising their hand. You can unmute and ask your question. Are you trying to ask a question? Then please unmute and ask a question. Xiaomi 2109.
yeah. Meanwhile, before Xiaomi can answer, in case
Dr. Abhijit, you're on mute in case you're trying to talk. Xiaomi, do you want to ask a question? I think it must have been done by mistake. We will take the two questions that are in the chat.
Okay.
Okay, we have Gaurav who wants to ask a question. Go
Yeah,
ahead.
yeah. So I wanted to ask. So you told that it's better to store in the vial. So is it like these are limited and these can't be replicated at a later stage?
see, Gaurav, that's your name, right?
Yeah, right.
Yeah, Gaurav. See, I'll tell you. Firstly, whatever number of cells you bank at the time of delivery, okay. Once the cells are frozen, I mean, this is something that in fact, it's something I'd like to emphasize for everyone here. Once you have stored them, okay, so basically once they've been frozen, you can do what you want. You can amplify them, increase the number, or add some growth factors. A lot of things can be done to improvise on the cells. But assuming everything is done, and once it's banked, which means once it's frozen, then it can only be thawed and used. I repeat, it can only be thawed and used. Thawed, which means it has to be brought back to the liquid state and then immediately transfused. After it's frozen, it cannot be thawed and then again improvised. So whatever number of cells you've banked at the very beginning are the only amount of cells in terms of quality and quantity and purity that you're being going to be left with for the tenure of your bank. So that is why, in case you bank, say, in 10 vials, those 10 vials are your custodian. So that is something that's going to be with you till you use it. You cannot thaw it, and at some other stage, at maybe a 50 years, you want to you want to improvise on those cells, or you want to amplify. That is not possible, because as soon as you thaw it, you have to use it for transplant only. You cannot add anything. You cannot improvise on the sample, and you cannot refreeze it, because even if you refreeze it, all the cells will die. So that is one of the benchmarkings of cord blood, basically any stem cells. You can do what you want before preserving or before freezing. And after you've frozen, once you've removed it from the repository, all you need to do is you need to thaw it, and then you need to use it. If you cannot use it, then you cannot refreeze it. So hope that answers your question.
Yeah, thanks for that. And I have another question. I've seen that most of the organizations which are working in this area, they are offering some kind of different packages. So they say that, okay, it's only the blood collection, or they will also store the, I think, umbilical cord, or I mean, you understand those medical terms more properly. But yeah, there are things, whether they will store it, whether they will only store the blood or they store some other cells as well. These kind of packages are there. What is the difference between them?
See, basically, products of conception can all be banked. At this point in time, Gaurav, there is only a license from the regulator for cord blood banking. Nothing else can be banked. There's no license for anything else. Secondly, ReeLabs also has a permission. In fact, it's the only stem cell enterprise, and I can say that on a public forum, that has the permission, explicit permission from the regulator to bank placenta. The only reason we have options or the permission to bank placenta is because we are doing clinical trials to showcase the application of placental stem cells in therapy, because of which we are banking placenta. And when you talk about placenta, because I'm seeing another question which is also similar to this, what is the difference between amniotic sac? I saw something like that: amniotic sac, amniotic fluid, and placenta. Basically, these are all ingredients of the placenta. Placenta is a universe. Okay, these are all subsets. So whether it's cord blood, cord tissue, amnion, amniotic sac, these are all subsets of the placenta. Placenta is a universe. So in case you bank placenta, you bank the universe. You don't need to bank anything else. Of course, cord blood will have specific hematopoietic cells which cord tissue doesn't have, but placenta has everything. So in case there's an option of banking all these products of conception, it's a good idea to take it. I also see a question on SLE. SLE basically is an autoimmune condition, which means your immune system is compromised, because of which it affects most of your organs and tissues. In this case, stem cells have a big role to play because it's immunomodulatory. That's exactly what it did in COVID also. It modulated your immune system, which means it improvised or regulated your immune system. So in case the immune system in SLE can be regulated potentially with stem cells, we can look at reversal of the condition. Of course, we have not reached the stage where it can be reversed yet, but are we galloping towards that stage? Answer is yes.
Thank you, Doctor. I think last question we have is, what are the best treatment options for CP? If you could just explain what CP is, because not everybody is in the medical field out here.
Cerebral palsy or CP is basically a condition where the child is essentially mentally retarded, and the mental retardation has happened because there has been some kind of trauma at the time of delivery, and there's been some kind of lack of oxygen to the brain, so that the brain has not developed during delivery. Something that can be avoided, but some things which happen without anybody's purpose or without anybody's fault as such. In this case, stem cells do have a role, because stem cells by default can give rise to any cell, tissue, or organ. That is the definition of stem cells. In this case, in cerebral palsy, the nerves are the ones, or the nerves of the brain are the ones that are compromised. So in case anything that can be done to repair your brain or repair the nerves of your brain, we are looking at a solution, we are looking at a cure. Thank you.
Thank you, doctor. Are there any more questions? Otherwise, we can end the session. In case any pregnant mamas, pregnant parents have not left their details in the chat box, please do so. We will be having a lucky draw, and the winners will get goodies from ReeLabs. Please leave your numbers in the chat box.
Okay, thank you so much for your time, and hopefully everybody's benefited from this session.
Thank you very much, Dr. Abhijit, for your valuable time. I am sure that we tried to get as many questions that are continuously asked by pregnant parents on our various support groups. We are always asked, and these are the questions that regularly, help them decide whether they wish to bank or not. So I'm hoping that this has helped everyone. And you can just leave your details in the chat box. We have left the details of ReeLabs in the chat box in case you wish to get in touch with us. And in case you wish for a consultation with Dr. Abhijit to discuss treatment options, he is a pioneer in the field of stem cells. He will be able to answer those questions. So please leave your numbers out here and we can get back to you. Thank you everyone for your valuable time. Thank you, Dr. Anuradha, for giving us this platform. Goodbye, everyone. Thank you for giving us your time.